Yes, ma'am.
A very good afternoon to all the dignitaries and participants who have joined us for this session 6 in our webinar series on fermentation technology. I welcome the speaker of this session 6, Professor Ramalingam, sir, Associate Professor, Center of Biotechnology, Anna University, to our webinar series Fermentation Technology. Now, I kindly request Dr. R.M. Erilersi, ma'am, Associate Professor and Head Department of Chemistry, Chairperson of Science, Guru Nanak College, Velachery, to deliver the felicitation address.
Good afternoon to all of you. Thank you, Dr. Bharti, for inviting me to offer my felicitations in this August gathering. I am proud to say that the Department of Biotechnology is an active department in our esteemed institution. The department keeps organizing various activities which are very useful to the student community and also to the teaching fraternity throughout the year. Even during this lockdown period, a number of activities have been organized to enrich the knowledge of all. Today, we have come to the last day of the webinar series on fermentation technology. Each and every one of us assembled have known about fermentation. In all our homes, it is an everyday process, as in preparing idli dosa batter, preparing curd, etc. But industrially, how fermentation is done in large scale, many do not know. The Department of Biotechnology has taken an effort to highlight this in this webinar series and illustrated the use of microorganisms and enzymes for the production of compounds that have application in energy, material, pharmaceutical, chemical, and food industries. We had listened to Professor Doble Mukesh, Dr. Samuel Jacob, Dr. Veena Gayatri regarding fermentation technology in food and pharmaceuticals, upstream process in a fermentation industry, media formulation for lab to industrial scale, types of fermenters, metabolite production, and mass transfer. Now, we have an eminent professor, Professor Ramalingam, from the Center of Biotechnology, Anna University. His subject expertise is in fermentation and bioprocess technologies. His lab in Anna University has a well-established fermentation technology lab with both pilot scale and industrial scale grade fermenters. His area of research is metabolic engineering in E. coli and Lactococcus balactis and also in biofuel production. I'm eagerly awaiting to listen to his talk on different modes of cultivation of microbes. My best wishes to the organizers of this webinar series, a special mention to Dr. Bharati and Dr. Mahendra Kumar. All the members of this department are highly efficient, and I'm sure that they will progress greatly and reach greater heights. My best wishes for this webinar series. Thank you, Bharti.
So thank you very much for your kind introduction, madam. And
Thank you, sir.
I wish the Guru Nanak College prosper in a very big way in biotechnology, and thank the organizers for giving me this opportunity to deliver a lecture on fermentation technology.
Now, I request our head of the department, Dr. Bharti Ravikrishnan, ma'am, to kindly introduce our chief guest to the gathering.
Okay.
Thank you, Mahendra, sir. Thank you, Erilersi, ma'am. Thank you, Professor Ramalingam, sir, for your warm words. It's my proud privilege to introduce the speaker of today's afternoon session, Professor Ramalingam, sir, to all the participants. He has started his career as production officer in fermentation in the year 1994 in JK Pharma Chem Limited, Kadlur, Tamil Nadu. Then he has moved on as to be an assistant manager in Synbiotics Limited, Vadodara, Gujarat. From 1997 to 1999, he has been the biochemical engineering pilot plant at Almbek Chemical Works, Gujarat. He has started his teaching career at Center for Biotechnology, Anna University, as lecturer from 2003 to 2008, and now he is holding the post as associate professor in the Center of Biotechnology, Anna University. Professor Ramalingam is a chemical engineer turned biotechnologist specialized in fermentation of genetically manipulated system in the bacteria, especially in the area of metabolic engineering. He has a well-structured industrial career spanning 5 years in 8 giant pharmaceutical companies, as mentioned above. As at present, he is working in metabolic engineering for the synthesis of green chemicals. He is also involved in the Marie Curie International Research Staff Exchange Program of Food Research Exchange Program between India and Europe and offered by European nation for metabolic engineering work package. This project involves collaboration in teaching and in research. Professor Ramalingam sir has completed various projects of worth nearly 1 crore 80 lakhs, and to mention a few: DBT Virtual Enzyme Center, development of enzyme formulations for the treatment of lignocellulitic biomass, multi-institutional projects, metabolic engineering of Lactococcus lactis for the production of propionic, jointly organized between Anna University and IIT Madras. Production of 1,3-propanodiol metabolic engineered lacto-retri using glycerol feedstock jointly between Anna University and IIT Madras. He has also delivered various talks in various nations, and to mention a few are he
Thank you,
has
ma'am. Thank you
delivered
very
a
much,
talk in
ma'am.
Department.
We'll cut short the introduction and go ahead.
Yes,
Yeah,
sir, sure,
thank you very
sir.
much.
Okay, you can start with your sharing your expertise with us, sir.
Yeah, sure, sure. So, good afternoon, listeners. It's a great pleasure for you to, for me to meet all of you, but it's not possible. But then I get connected through this online mode. And today I'm going to talk about different modes of cultivation of microbes. Perhaps these different modes of cultivation can be used for bacteria, for animal cells, for plant cells, for any kind of unicellular organisms. But today, anyway, I'm going to focus on the bacterial part of it. And yeah, so all these cultivation modes can be carried out in this reactors. And I'm showing you the picture of a continuous stir tank reactor. There are different kinds of reactor, but we can use the cultivation mode in any type of reactor, but the one which is most widely used is this continuous stir tank reactor. It's the workhorse of the industrial world. Therefore, I'm showing you the picture of the laboratory scale bioreactor here. And then it's a large scale reactor where you can see from the top, there are all the lines to the reactor connected from the top of the reactor. And this view is the internal view where you can see the impellers, agitation devices, which is used for mixing the fermentation contents. Actually, this is a baffle which will prevent the vortex formation. And these are open reactors, very large reactors where they have kept it in the open, where there is no covering. So this is the structure of the reactor. Perhaps you might have studied or seen it in different lectures. But here today, how we are going to use this reactor to produce product by using various cultivation modes is what I'm going to talk about. And one of the major goals of this fermentation technology is to maximize the product. We want to make more amount of product that would result in maximum revenue to the industry. And this maximum product formation can be accomplished by two means. One is that you can make every cell, be it the product is intracellular, be it is extracellular, the only way is that you can make every cell to produce more product. That is very important. And secondly, we wanted to house or accommodate as many number of cells as possible. So that's what we look for. By doing these two things, we can maximize the amount of product formation in the reactor. And hello, can you hear me?
Yes, sir. Yes, sir. We are able to hear you, sir.
Yeah. So, the product can be maximized by these two things, and we need to accommodate these cells inside the reactor, which means that we can't dump them like in a town bus or in a metro rail. We need to provide them comfortable accommodation in the sense they have to work very efficiently to produce the product. So in that regard, we need to look into the oxygen supplying capacity of the reactor, the heat removal capability of the reactor, and how efficient they can mix the contents. All of these aspects have to be taken into consideration, and thereby we'd like to achieve very high amount of titer, that is high concentration we want to achieve, because the higher the concentration, the reactor size required will be smaller, all the downstream processing equipment's size will also become smaller. The volume of extractants or solvents used for purification, everything will become lesser. So operating costs will become lesser. Then only it will become viable. Likewise, the rate at which the product is formed is also very important. We need to have a high product rate, that is around 1 to 2 gram per liter per hour of product should be produced. And maximum amount of the glucose or whatever carbon source you are giving, that should go into making the product. That is what we call it as yield. What fraction of the glucose goes for making into product, rather than making biomass, rather than making carbon dioxide and other byproducts. These three things are very important. And in order to achieve the maximum of these three things, we tried different cultivation modes. So how to make every cell to produce more product? As I've told you in the beginning, it's very important. There are two ways by which you can make every cell to produce more product. One is that you can develop a high yielding mutant by random mutagenesis. And second is by metabolic engineering. So by doing this work, you can obtain a strain which is highly evolved to produce a particular product. So what are the various ways by which the mutation is done? So the mutation is nothing but they do random, they mutate the cells, millions and billions of cells, and screen for a cell which produces more product. And by that way, they can evolve a better producing strain. And in the process, what happens? For example, we want to make a lysine overproducer. From aspartate, the lysine will produce along with methionine, isoleucine, threonine, for example. And if this threonine isoleucine is produced in a certain amount, it goes and binds to the enzymes in the pathway, and it exerts feedback inhibition. And when you want to overproduce one particular metabolite, you need to knock out or engineer the protein such that these feedback inhibitions, for example, this lysine will go and aspartate kinase, it will try to inhibit. So we need to remove the inhibition so that the more amount of aspartate will flow into the pathway. So this is how it happens by random mutagenesis. If it's going to be metabolic engineering, for example, we want to overproduce succinate from E. coli, from glucose in E. coli, which means that it also produces lactate, it produces acetate, ethanol, and we need to knock out all these genes. By doing all these genes and overexpressing pyruvate carboxylase, then you can produce more amount of succinate. So by these two major strategies, scientists are able to develop a strain which can produce more product. Once that strain is available, then the next most important thing is to provide the right physiology of the organism, which is essential for making the maximum amount of product. So how do you provide the appropriate physiology? That is, we need to provide a particular pH, temperature, dissolved oxygen concentration, nutritional composition in terms of carbon, nitrogen, and also inducers. Sometimes it requires inducers, sometimes it requires amino acids, sometimes it requires lack of these amino acids. Like that, the requirements depends upon a particular fermentation. And we also have to control the nutrient concentration at a particular levels. All these things have to be done. And more importantly, or most importantly, we should be able to control the growth rate. The growth rate, that is the speed with which the cell doubles, gives you a particular physiological status to the organism that results in maximum product formation. And the next few slides, I'm going to show you how the physiology of the organism changes with the growth rate, with the various conditions like temperature, pH, when it is growing and when it is making product. We can see lots of changes. For example, this pie chart indicates that the various proteome levels for growth, nutrient readiness, stress, for example, stress resistance, regulation, all of these things, this composition is widely varying. You can see from the picture. When the cell is growing at a higher growth rate, the more of the proteome is allocated for growth. When at a very low growth rate, it is rather hoping to get other nutrients and preparing for using the nutrients. And also it is preparing for stress resistance because it may undergo starvation. Likewise, at different temperatures, for example, you want to cultivate the bacteria at 28 degrees, E. coli, for example, at 37 and 45. And you can see when you're going to grow at 45 degrees centigrade, molecular chaperones are produced because it undergoes stress response, cellular stress response. To overcome this response, where the proteins will get aggregated, chaperones are produced to fold all these enzymes in a proper functional form so that cell can act properly or conduct the physiology. Is a metabolic pathway wherein at two different temperatures you can see that the red indicates very high flow of metabolites in the pathway and green rather less. You can see that at 28 degrees centigrade, all the pathways showing red, but a fraction of it is showing green, which means the amount of metabolite flowing in that pathway is minimized. And also I'm showing another example where you have two different conditions. One is that we make the cell to Corynebacterium glutamate to grow, and a second condition where we are making this Corynebacterium glutamate to produce high amount of lysine. And this is the EMP pathway, this is the pentose phosphate pathway, this is the TCA cycle. And you can see that the thickness of the arrow, thickness of the line indicates the amount of carbon flowing into various metabolic pathways. And you can see that the one for growth is different from the one for product formation. Here the pentose phosphate pathway is very large, here it is very low because it needs NADPH for lysine synthesis. It is made only in this particular pathway. Likewise, it respires, it makes more carbon dioxide when it's growing. So the TCA cycle is very more prominent. But here it allocates most of the carbon dioxide for lysine synthesis. So the amount of carbon flowing in the TCA cycle is less. And also the carbon flowing in different fraction of the cycle is also different. So which means all these indicate this genomic analysis, transcriptomic analysis, proteome, and metabolomic analysis. All of them, they indicate that the physiology of the organism changes with all these conditions, temperature, pH, no growing or non-growing conditions. So we need to provide them. So the most important thing is to provide all these favorable conditions for product formation. And as I've told you, controlling the growth is one of the most important thing. And the speed which the cells multiply, okay, I'm going to call it a specific growth rate, which is the inverse of doubling time. That is, when the cells grow faster, the doubling time becomes smaller. Doubling time becomes smaller. And this is a graph which shows that as the growth rate, the speed with which the cell multiplies is a function of the concentration. As you increase the concentration, the cell will start to grow faster and faster. And beyond a certain concentration, say even 1 milligram per liter for glucose, it can grow at the maximum speed. And after that, whatever level of concentration you provide, the cell cannot increase the growth any further. They'll be growing at the same mu max, we call it as maximum specific growth rate, or the shortest possible doubling time. And so based on these two knowledge, that is by trying to provide the favorable condition and also by controlling the growth rate, we can provide the required physiology that will result in maximum product formation. Secondly, we need to increase the cell concentration. That's very important. Only then we get higher product. And also we have to continue this fermentation for as long as possible. So these three things when we do, we get high titer, rate, and yield. So to not accomplish this, scientists have developed, there are various cultivation modes. Today I'm going to talk about 4 different cultivation modes. One is batch, the other one is fed batch, the third one is cell retention cultivation, and the fourth one is continuous cultivation. We call it as chemostat. And also two chemostats are used in tandem. Then we call it as 2-stage chemostat. Let's first go to the batch cultivation. So how is batch cultivation done? So this is the reactor, and we either sterilize the reactor without any medium, then we can add a sterilized medium using a sterilizer, continuous sterilizer for example, or we can add the medium and sterilize it. We add the carbon source like glucose, sucrose, nitrogen source, organic nitrogen like constipulaca, or say cas amino acids, or ammonium sulfate, urea. They are all inorganic nitrogen. We add buffer, macronutrients like magnesium, calcium, and also trace metals and also antifoam. We add all of them. We can't sterilize all of them together. We need to sterilize carbon source and nitrogen source together. Otherwise, they interact and they are not available for the cells. So even trace metals, we have to sterilize them separately. A few of the components have to be sterilized by filter sterilization. But all said and done, we need to fill up the reactor with the medium, either sterile or non-sterile. Once it is sterilized, then we need to inoculate. You need to inoculate the reactor by using this inoculum, which is also a small reactor, which is prepared in a small reactor. And in a small reactor, they'll add it from the shake flask. And then we will set the conditions, parameters like aeration, agitation, pH, temperature, dissolved oxygen values. All of these values have to be set. And once it is done, then the fermentation will start. And usually I'm showing a graph of how the cells grow. So the cell will grow logarithmically, we also know that. And then for two different conditions, two different glycerol concentrations, they have done these experiments. But in all these cases, the glycerol will get exhausted and the growth will proceed. When the glycerol gets exhausted rapidly, the growth will be faster. When the glycerol gets exhausted slowly, the growth is slow. But all said and done, it is growing logarithmically. So the batch cultivation lab, lag phase, log phase, stationary phase, and death phase. And in industrial world, lag phase they don't want because they don't. It's not a productive phase. So we add sufficient amount of inoculum. We take logarithmically growing culture. We also have the same medium composition between the inoculum and the reactor, so that there will be no lag. Straight away, cells will start growing from log phase. And once the nutrient get exhausted, it will hit the stationary phase. Then the rate at which the cells are produced will be equal to the rate at which cells die. And then after some point, the cells will start to die. So death phase will set in, but we don't go to that point. We'll stop here. So there are two kinds of products we can make by this batch cultivation. One is the growth associated product, and the second is the stationary phase associated products, non-growing associated product. And one of the major drawback with this particular batch cultivation is that, as I have stated earlier, when the concentration of the substrate is more than few milligrams, the cells will grow only at one speed, that is maximum growth rate. So that when they have added 30 grams per liter of glucose or glycerol, the cell will always be growing at one specific growth rate. The doubling time will be only one. It cannot be many. So that you cannot fine-tune the cells to grow at a particular speed with which you wanted, that will result in maximum product formation. Like we organize a cultural festival, what do? We distribute t-shirts to people. And what do? Do we have different sizes in that? No, because we are producing on a very large scale. We want to avoid confusion. So we have three sizes, L, M, and small, middle size and large size, so that all of you have to fit into it. So that's not a best way of wearing the nice t-shirt rather than looking for the most appropriate size. So similarly here, there are there are products which will be produced at a very slow growing phase, but we cannot provide any of them. All the slow growing products which are produced under slow growing conditions will be produced during the stationary phase, and all the growth associated products, whether at a low growth rate or high growth rate, they will be produced only at the maximum speed, which means each and every cell will not be functioning optimally. That's one of the major problems. The second major problem is that, as I've told you earlier, we need very high cell mass so that every cell will produce more product that will result in high concentration of the product. That's very important. In order to do that, when you want to increase the cell concentration, you need to add more and more substrate in the beginning of the batch process. When you do that, for example, when the substrate concentration increases, it results in hyperosmotic solution kind of a thing, where the cells will shrink, and it is called plasmolysis, and cells don't function effectively. That is the reason why we have very concentrated syrup at home. Say gulab jamun we are making or rasgulla you are making, those sugar syrup never gets contaminated. It need not be refrigerated. It can be kept at the room temperature. That's because of the very high concentration of the substrate. That is sucrose there. Sugar is a highly concentrated in that. If you dilute it, it will get contaminated. So that problem we'll face. Substrate inhibition is a major problem, and also another high concentration of the rapidly utilizable glucose or nitrogen sources there. It goes and represses the product formation pathway, and that we call it as catabolic repression. So product will not be produced. That's another major problem. The third major problem is that when you want to cultivate large number of cells, if they grow at the maximum speed, their oxygen, each and every cell will consume huge amount of oxygen, so that the reactor will not be able to supply oxygen when you have very large number of cells. So only way you can have high cell concentration is that by allowing them to grow very slowly, so that the amount of oxygen consumed, the amount of heat liberated will be under control, so that the reactor can supply oxygen, remove heat. So that's a very important thing. For these two major drawbacks, that is, you cannot maintain a particular specific growth rate that is essential for maximum product formation. You cannot increase high cell concentration. Both important things cannot be done with this batch cultivation. So scientists usually don't prefer batch cultivation. We'll start with batch cultivation for all basic optimization studies, but we always go for other cultivation methods which are much, much superior than the batch cultivation. So when you have substrate inhibition, then the best way is fed batch cultivation. Why? Because in this fed batch cultivation, initially we will start with a very small amount of substrate. We'll put into the reactor, we'll start as a batch, inoculate. Once the cells grow, the substrate get exhausted, a certain amount of cells would have formed, and then we will start feeding the biomass, feeding the reactor, like very concentrated carbon source, nitrogen source, only sometimes carbon we will add, and then little by little the volume will also increase. Cell concentration also will keep increasing. But when as much as we feed, the substrate will be immediately consumed by all the cells there, so that the substrate concentration will never increase in the reactor. So whatever low concentration of the substrate you can start, the batch will be very small. Batch can be very, very small. And then once the substrate gets exhausted, we can start the feeding. So here, by this way, the substrate concentration will be kept under control, so that substrate inhibition problem is overcome in fed batch, and also you can get a very concentrated product. So I want to show you an example of how we do an E. coli fermentation, recombinant E. coli. We will start like a batch. Like I told you, biomass will be formed, substrate will be consumed. Then one substrate is exhausted, then start feeding. And one cell will become two will become four, and they're going to go exponentially. At every time point, every cell should receive the same amount of nutrient. In order for that to happen, when the cell is growing exponentially, we also have to feed exponentially. When 2 cells are there, we're going to give them 4 units of substrate. When 4 cells are there, we're going to give 8 units of substrates, like that. The feeding also have to be matching the growth rate at the speed with which the cell is growing. And secondly, by controlling the substrate concentration during the feed, you can control the growth. So by controlling the growth, you can control the product formation. And usually in recombinant E. coli, we will build the cell mass to a certain extent, such that whatever is possible in that reactor, based on the oxygen supplying and heat removal capacity, and then we will induce the cells, switch on the gene expression to produce the product, and the cells grow again. They again continue to feed during the product phase, also product formation will result. By that, we control the specific growth rate that results in maximum product formation. The same thing holds good for antibiotic. So all non-growth associated products are very well produced in fed batch. The reason being that it is not the stationary phase, which is actually the best phase, but slow growing phase. It requires a very small amount of nutrient, but they require them continuously. That is what we provide. Initially, we provide glucose, concentrate liquor, soybean flour, cottonseed flour, ammonium sulfate, phosphates, everything we dump inside, start as a batch. They grow rapidly, and once nutrients get exhausted, Then we will feed glucose. We feed glucose and ammonium sulfate in a controlled manner so that the cells will grow very, very slowly, and antibiotic will start to get accumulated. So we can all, so that therefore in fed batch cultivation, two advantages are there. One is that we are able to increase the cell density, we are able to control the specific growth rate, the doubling time we can control, which is very crucial. So by doing these two things, for a substrate inhibited fermentation, fed batch cultivation is much superior to that of the batch cultivation. And when you have product inhibition, what can you do? As in fed batch, your product concentration is building up. If this product is going to inhibit the growth, it is going to kill the cells, then what shall we do? Just like ethanol fermentation. Ethanol, when the concentration reaches 6 to 10 percentage volume by volume, 60 grams to 100 grams per liter, it kills the cells. So you cannot increase the concentration. In that regard, then we have to go for cell retention cultivation, wherein we will pass the cells through a microfiltering membrane. The cells alone will keep cycling inside, and the medium will be removed. The medium will be continuously removed so that the product concentration will not increase. So fresh nutrient have to be fed continuously so that the volume will remain constant. So either we can use a microfilter or we can use a centrifuge. So the cells will be centrifuged and will be cycled back into the reactor. Only the clear medium will come out along with the product. Fresh medium will be added, but medium loss will be there. A lot of huge amount of medium will get lost, which is unused, but you can't help it. But you can get product out of it. And if you completely recycle, it will act like a batch cultivation. Cells keep on increasing, then you have to stop. But you can also partly remove some amount of cells, then you can continue the fermentation. So the advantages with this retention is that suitable for product inhibition, specific growth rate can be controlled because you can control the feed rate, you can control the feed. When you control the amount of substrate you're feeding, then the rate at which it is growing, it can be controlled. And also high cell concentration can be achieved because the cells are not going out, it's getting recycled. But the problem is that cells will starve of oxygen in these circles, so they try to give air, but that's not a good thing. And also the cost of membrane, centrifugation cost, they're all expensive and cumbersome. In a very large scale reactor, this is not very interesting. So that is these are drawbacks. And this diagram I want to shift. So the best method for getting the maximum output continuously from a reactor is continuous cultivation. Here I am showing a popcorn machine wherein the corn is continuously fed to the machine and the popcorn is continuously thrown out. And normally you don't get to see that. They dump all that corn into the stuff and it makes popcorn, and it's a batch process. And every now and then you have to stop the batch, start the batch, do cleaning, do harvesting. Sterilization takes time, and also when the cell is growing, it doesn't produce any product. So only a very small fraction is very meaningful. That is productive. All the other phases, the product doesn't come out of the reactor, so that the amount of product that is thrown out of the reactor is always lesser in all the other cultivation modes compared to continuous cultivation. But the major drawback with this continuous cultivation method is contamination. To start and get to the steady state, it takes a lot of time. It takes 3 to 4 days to get the steady state. And the rate at which the cells are formed in the reactor will be equal to the rate at which the cells are removed. And this is the continuous cultivation arrangement, wherein we continuously feed the nutrient and continuously remove the medium along with cells. So cells also get lost. They spend a very short time here. That's the problem. You cannot build cell concentration here. That's another problem. The only advantage is that the size of the reactor, for a given size, continuously the product can form for years if it is a stable process. It is very suitable for product inhibition, and we can get the maximum productivity when compared to any other cultivation method. But the major drawback is the contamination problem. And also the second major problem in continuous cultivation is the reverting of mutant to wild type. I have told you already Cells are genetically engineered or mutated to produce high amount of product. We all know that there is a gene repair mechanism inside the cell. They constantly repair all these mutations. So, father will do some mutation, son will do some mutation, grandson will do some repair, more repair. So when more and more generations, you can see that all these changes are reverted back. They repaired all those mutations and they become wild type. They happily eat the substrate. They grow happily. They don't make any product. There's a major problem. And the third problem is that the conditions optimal for growth is different from product formation. For example, here in antibiotic fermentation, the fungus grows very well at pH 5.7, but it produces product maximum penicillin only at pH 6.5, 6.6. So here we set the pH at 5.7, 5.5, or even less, allow the fungus to build up. Once enough biomass is made, we shift the pH to 6.5. And here we make them grow rapidly. Here we make them grow very slowly. But you cannot do that in a single reactor. Two different conditions, just like co-joined twins. They can't. One twin has to, keep in tune with the likes and dislikes of the other twin, and that's how they can live their lives. The same way, two different conditions cannot be provided in the same reactor. This is another major problem, and all these problems are overcome in one situation. That is, they're trying to produce single cell protein production by using methanol. So methanol, no other contaminant can grow. So contamination problem was solved. It's just a cell, single cell protein. It is not mutated. So there's no reverting back. And also, since the product itself is cells, there are no two different conditions. So in England, 25 years back or 30 years or even 40 years back, they produced single cell protein on a very large scale by continuous cultivation. Other than that, there are very, very less continuous cultivation processes that are prevailing in the industry. But nevertheless, this continuous cultivation is a very good tool because you get a steady state. The rate at which the cells are formed in the cells will be equal to the rate at which the cells are removed. And by controlling the feed, you can control the doubling time. So that at different, you can control the doubling time of the cells, and this doubling time can be maintained for a very long time, so that you can understand the physiology of the organism very well in this continuous cultivation method. So to understand the cells, we always work with chemostat in the laboratory. So it's a very good scientific tool. And when you want to overcome this problem of two different conditions, you can have two different chemostat. One can be used for growth, the other can be used for product formation. It is also done like that. But then even one reactor is a contamination problem will be there. Two stage in a very large scale, industries don't prefer. So to summarize, so all these different cultivation methods are deployed based upon our requirement, based upon the kind of product we are looking at, and what is the cost of the product. So based upon which the best cultivation strategy is chosen. And I have not gone into how to design these strategies, but I have just described only the rationale behind it. But if you understand the rationale behind it, and in future, if you take more interest towards it, I think you can also learn to design the cultivation processes. Thank you. Hello? Can you hear me?
Yes, sir. Yes, sir. We are able to hear you, sir.
Yeah, fine. I'm done. I'm done with my lecture.
Yes, sir. Wonderful.
Thank you, sir. Thank you so much for enlightening us about the various modes of microbial culture, sir. Now I kindly invite Dr. Mahendra Kumar, Assistant Professor, Department of Biotechnology, to take over the discussion session.
Thank you. Thank you very much, sir. Thank you very much, sir, for the wonderful and insightful presentation.
Thank you, sir.
Just pop up some of the questions asked by the participants.
Yeah.
Dear participants, I request you to kindly type your questions in the chat box so that sir can answer.
They can't speak actually.
Sir?
Yes, sir.
So they can talk also. They can come online and ask questions.
No,
What is that?
no, sir. In YouTube, they can only type their questions, sir. There
Okay.
is no option for us to unmute and speak. It is not like that, sir.
Okay.
Sir, is it
Okay.
visible,
Yeah.
sir?
Yeah. How to maintain steady state in a continuous cultivation? Yeah. That's a very good question. And once we decide on the rate at which the cells have to grow, then we will decide on the concentration of the nutrients which we are going to feed, and also the rate at which we are going to put the substrate. So the rate of feeding will decide the rate at which the cells will grow. So you can start as a batch and then you start feeding. And after a period of few, say 12 hours or 13 hours, there is 5 volumes of medium have to flow through the reactor. Once it is done, then automatically it will reach the steady state. And the rate at which the cells are generated will be equal to the rate at which the cells are removed. The number of cells in the reactor will remain constant. That is how the steady state is maintained. So you will get the steady state. So, yeah, Aarti Raval, thank you for your question. Please give some details on cell retention cultivation. Yeah, what details do you want? We actually cycle the cells through the microfilter and put the cells back into the reactor because we don't want to lose the cells. And in the continuous cultivation, for example, cells are also continuously removed. So the cell concentration you cannot build up. But in the case of retention cultivation, the cells alone will be put back into the reactor and only the medium will be removed so that the cell concentration will keep increasing, and we can also maintain a high cell concentration. And if you remove a small amount of cells, depending upon the rate at which the cells are growing in the reactor, then you can maintain a chemostat or continuous cultivation like arrangement and also maintain a high concentration. Steady state will be achieved, and then we can make more product. The third, I'm sorry, the third question they are asking is Madan Kulkarni, how to collect algae from agar tube culture. Uh, yeah, this you have to talk to a microbiologist, how to really work on algae culture. And how do we manage to add chemostat or turbidostat on large scale? Yeah, turbidostat is one. We simply measure the turbidity of the cells. We maintain the cells constantly and we feed the nutrient. But the turbidostat is done at a specific growth rate near mu max. But chemostat is one where we control the growth rate by using a limited amount of substrate. Yeah, thank you, Tarun Bure. Yeah, for secondary metabolite production in fungi, what precautions should we need to take? For, for secondary metabolite production, first you need to grow the fungus, allow them to build up the required biomass. So if you add too much of glucose in the beginning, it will grow fully and it will become very viscous. Fungal fermentation will become very viscous. So you need to add the appropriate amount of glucose and nitrogen sources so that the growth stops at the right point. Then you have to feed little by little. Then you have to make the fungus to produce a secondary metabolite. That's the most crucial thing. Thank you, Nilima Devi. What about the process economics in these three types? The process economics will be depending all the three types. Fed batch is more economical amongst all these processes. Batch is also economical, but fed batch will be the best. Yeah. Which cultivation will be ideal for fungal drug metabolism for impurities? I can't really understand drug metabolism for impurities. I'm not very sure about it, but you can do fed batch cultivation. That's the best for even analyzing the impurities or even drug product formation. Yeah,
That's all, sir.
Srinivas, which cultivation will be ideal for fungal drug metabolism? Certainly fed batch.
Okay, sir. Thank you very much, sir. I
Fantastic.
think more than questions, there are a lot of appreciation and thanks message, for making them aware about this. And one more question, sir.
Yeah. How can I increase the cell biomass of Bacillus coagulans and stimulate sporulation? Yeah, bacillus coagulations. Perhaps you have to work at a higher pH. I think I'm not very sure about it. Probably 9 in the range. You may have to grow around 7 to 9. Find out which pH it grows well, and then you go for fed batch. You start with a small amount of medium, then you start feeding the medium, design feed, then increase cell mass. That's how. And once nutrient get exhausted, allow it to starve. Starve for 12 hours to 14 hours and check for the spores in the microscope. And then you can see the formation of sporulation. Once it is completely sporulated, then that's how you have to harvest. Thank you, sir. Thank you. Welcome. Welcome. Ragini Bodhi. Yeah.
Thank you very much, sir, for answering all the questions raised by the participants. Now I
Yeah.
request, yes, sir.
Shall we wait for a few more minutes? Any more questions will come?
Yes, sir. That's sir. We can wait,
Yeah.
sir.
Yeah. We give them time. I think answering is very important to the
Yes, sir.
participants. They can ask any kind of questions. Participants, feel free to ask any kind of questions. Yeah, they are economical. They are economical, definitely. When we make any product, the cost of manufacturing should be less than 30 to 40% of the total cost of product. That's how we work. So welcome, welcome, welcome. Yes, madam. How can we prevent contamination? Wow. This is one of the toughest questions, Darun Murray. Excuse me. It's a very tough question, and we struggle very hard in industries to maintain contamination. We sterilize the whole building for that we go to that level. And the best way is to ensure that all these O-rings, where there are joints, there will be gaskets and O-rings, they have to be ensured they're intact. That's very important. And all the sterile technical operations have to be technically done properly. That's very important. How to separate viable cells from dead cells? It's pretty difficult. And there are methods being developed by people for, say, for Pichia pastorists. They are developed in our laboratory. They are working on trying to develop viable and dead cells, but I'm not very much aware of it. Control the pH changes immediately. Yes, we can control. Yes, we can control the pH very well. That's not a problem. In fed batch cultivation, pH control is either by feeding or by adding acid or alkali, we can control them. Yeah, India has fantastic reactors. I have operated India has some of the largest reactors, 2 lakh liter reactor. We have lots of reactors. There is so much of scope for fermentation technologies. Every year they're asking for people for with the skill set of fermentation. Algae as SCP can be best cultivated in open ponds. Open ponds as of now is said to be the best because you cannot increase the cell concentration because light shielding effect is there. That's the major problem with algae. Open pond is the best if you want product, but the product is high value, then you can go for reactor. Otherwise, for biodiesel, you cannot use algae in the reactor. Thank you, sir. Thank you very much.
Participants, you can ask your questions. Sir will be. Sir, I think sir is very eager to answer.
Yeah, that's very important. Any kind of questions you can ask. So the session where we are here to give you the guidance, and you can ask any kind of questions. It may be ridiculous as well. It may be irrational. Doesn't matter. Ask questions, which I may not also be knowing. Nothing wrong in it. Which is the best organism for a single cell protein production? I'm not very much sure as of now, which is very best. Methlotropis methylophilus was used earlier. Even today, you can grow any of these organisms by using methane or any other easily available cheap raw material, and that's the best organism. Welcome. Thank you.
So shall we conclude, sir?
Yeah.
I request Dr. Bharati Ravikrishnan, Head Department of Biotechnology and the convener of this webinar series, to say the vote of thanks. Madam, please.
Thank you, Mahinder sir. It is now my proud privilege to render my vote of thanks for the session 6, a part of the webinar series on fermentation technology. It is my foremost duty to thank Professor Ramalingam sir, who has readily accepted our invitation and to deliver his talk today. It is indeed our privilege to listen to you, sir. I place on records my heartfelt thanks to our General Secretary and Correspondent, Mr. Manjit Singh Nair, for supporting us in all our departmental activities. I thank my principal, Dr. N.G. Raghunathan sir, both Vice Principal, Dr. LRS Kalanidhi ma'am and Dr. Gunita Arun Chandok ma'am for their constructive suggestions. It is my foremost duty to thank all our participants all over country who has joined us during this session. I take a moment to thank my department colleagues, Dr. P. Mahalakshmi, Mr. Amal Ludraj, Dr. Mahinder Kumar and all my students for your support and contributions. Thank you all. Now I request Mahindran sir to post few instructions.
Thank you very much, ma'am. Dear participants, the feedback link for this session 6 has been posted in the chat box. Kindly submit your feedbacks, and this YouTube URL for the session 7 to be held tomorrow will also be sent to you by WhatsApp and to your registered email address. Kindly join us for tomorrow's session, and we need your support for that too. Thank you very much.